Yes, good morning.
Good morning, sir.
Morning, sir.
Good
Yeah,
morning, sir.
sorry for the delay.
Good
There was
morning,
one
sir.
yeah, good morning. There was a delay because one of the student is facing problem in his exam, so I was trying to sort it out. So fine. So today we have around 21 students on board. So let's begin. So yesterday we started with the topic called as an metabolomics. Okay. So there are few things which we already discussed. So first let me present the screen and then I will explain it again and then start with it. Yeah, is the screen visible?
Yes, sir.
Yes,
Okay,
sir.
fine. Yeah. So that is what we started with metabolomics. So this is what I have explained you. I think you got an idea about this, that about a particular cell when it is in a normal conditions. Okay. So it may have the functioning like this, that is DNA to RNA, RNA to enzymes, and then metabolism, whereby the nutrients are taken in, the metabolism will take place, the energy will be obtained, the biomass will be obtained, which is required for its normal growth and development. Fine. And some of the nutrients which was taken in it could be excreted outside the body, and that is what has been studied or which is being happening with respect to the classical metabolic engineering. Fine. So um, this is what I told you about it. Yeah, this is also done, but then with much more progression into the different type of the technologies or the instrumentation uh, and reverse engineering came upon, whereby if you really want to go and successful maybe genetic modification or engineering a particular organism, you should have to think in the reverse way, because it is the metabolism which is not metabolism but the metabolite which is playing an important role. Right. So first you will have to identify a metabolite, then identify which metabolic reaction it is involved, what is the actual reaction, what is the enzyme which is required, what is the RNA which is producing that enzyme, and what is the DNA or the gene which is responsible for that uh, RNA. So this way you can go in a reverse way and try to find out the specific gene and target only that specific gene. And that is what is constituting the modern metabolic engineering, where we are trying to go for the reverse engineering as compared to the classical metabolic engineering. Right. So there are few principles which I have already discussed. So that is what I told you. Then, if you go for a successful metabolic engineering, I told you that there are four steps which we have to follow. That is what DBTL. That is design, build, test, and learn. Right? Means you'll have to decide upon your metabolite of your interest or the change what you want. Okay, so it could be a phenotypic change, and this phenotypic change is brought about by a change into the concentration of your metabolite. Say for example melanin, if I say that if there is a change into the concentration of melanin, obviously the color of that human being is going to change. Right. So you target your metabolite. Fine. And then you target which host you are trying to work upon, whether it is going to a prokaryotic system or an eukaryotic system. Now we know both the systems, their complexities, so based on that you can think upon your metabolite, its pathway, and the host in which you are trying to do the modification. So that is what is required into this. Then you go for um, understanding the whole mechanism, that how that metabolite is produced, what is the actual reaction, what is the protein which is essential, and what is the gene which is essential for that. Okay, then you try to study the genetics of that particular gene. Means what kind of transport what kind of promoter is required, what kind of operator is required, are there any some regulatory elements, etc., etc. You try to study that, try to modify those particular genetic elements for the production of that particular gene. And the most advanced which I told you, that is the CRISPR Cas which can think of. So you try to work upon these genetic sorry the editing tools, So it may involve the molecular biology techniques, it may involve the genetic engineering techniques, etc., etc., which will be supplemented by the bioinformatics things, right? So maybe whatever kind of modifications you are thinking of, you first try to do it into a dry lab using the bioinformatics tools. And if you are successful in that, then you think of going for the wet lab. Okay, so this is what is considered into the build. Then you go for the testing, that you have done the modification. Okay, then you try to find out whether the output what you are expecting, you have got it or not. And for that, as I told you, you have to go for the high throughput screening. Means you may have to experiment with n number of samples. Okay, and you cannot go experimenting in means one by one the samples you can go for the analysis. So it should be a case that at a given time thousands and thousands of your samples should be able to analyze, and that is what nowadays we have the sophistication into the instrumentation. So that is called as an high throughput screening, which could be supplemented with the smaller analytic techniques what we have. It could be the spectroscopic, the chromatographic, the microscopy, etc., etc. So with all these available technologies, you can go for the testing your system. And once your testing has been done, whatever the data you get, that data you'll have to understand that what actually happening into the system. Okay, and one of the thing what we usually do when you are studying the metabolomics or the genetic engineering, sorry, the metabolic engineering, that you try to understand the metabolic flux. Now flux is what? It is the rate at which that particular metabolite is formed or it has been utilized, right? So we go for the metabolic flux analysis, we go for the data interpretation. So with all these things, at the end, if you come to know, yes, that whatever the output you have thought of, you are reached to it. Maybe you may come to an completion of your experiment, but if you find there is a minor variation, if the things are not working, again you will have to go back to the design. That is, you again give a thought upon it, that whatever you started with, whether that thinking or that hypothesis what you have laid down, whether it was really correct or it requires some or the other kind of modification. So try to do that modification, then again go for build, then test, and then learn. So this cycle may, you may have to repeat n number of times unless and until you give you get the desired product in which you are interested, right? So this is what is required for the metabolic engineering, right? And this is what we are going to study in detail. Okay, here you have to consider cell as a factory. So there is something which comes inside the cell, that something could be in the form of a nutrients, it could be a carbon source, fine. So that carbon source, it enters, it undergoes a typical main metabolic pathway which could be having many branches. So you have that nutrients which are converted into the different uh, carbon forms. Okay, and when this conversion is taking place in the different carbon forms, you are getting the energy, fine. So you are getting an biomass which is generated, you are getting an energy which is generated, and both the things are required for the growth and development or the different activities of the cells. And few of the things, as I told you, could be excreted as an output, fine. So it could be an waste material, or it could be some important metabolite which is being produced. So it could be an antibiotic, it could be an acid which is being generated, or it could be an ethanol, alcohol, in case of the anaerobic what we may talk about. So that kind of output could be brought about. So you can imagine a cell as a factory, because in a factory we have the raw material which is coming in, it is undergoing the different processes, and at the end what you get is an output. Now, along with the output, you may get some waste material also. So that is what could be a synonym example for your cell as an factory. आलं लक्षात? The metabolic engineering, what we are trying to do, could be thought as a directed evolution strategy. Now, all of you know that how evolution has taken place. Maybe when the life it came into existence, what we saw, that there was an continuous change into the environment. Okay, means when I'm talking about environment, you can think about the biotic as well as the abiotic factors, right? So whenever there is a change into the environment, the cell has tried to modify itself. Okay, it has tried to accommodate in that particular environment, and for that accommodation, what the cell has done, it has done some modification within itself. Now that modification could be at the genetic level, right? So it might have modified itself in one particular gene, which could have been resulted into the production of some specific type of the proteins. Okay, and those proteins might have been responsible for production of some specific metabolite, which has allowed the organism to grow in that change into the environment, right? So evolution is what? It is nothing but an modification process which has taken. So maybe say suppose the first cell was generated somewhere in desert, in a hot area, but if the cell it has moved towards the polar condition, so obviously it will have to accommodate itself to that cold condition. So maybe in that particular environment, the cell has done some modification where they have produced some specific proteins, some specific metabolites, which has helped it to live into the very cold condition. Okay, so this is an typical example which I have quoted here. So when you're talking about evolution, what you can say, it is an engineering which is done by the nature. Okay, although this evolution or this engineering by the nature is a very slow process, but still it is doing the modification. Okay, now this modification what the nature is doing, you can think that it is a random type of an modification. Okay, but then when you're talking about metabolic engineering, we are targeting a specific protein, a specific metabolite, a specific gene. So that is why we can call metabolic engineering as what? A directed evolution strategy. Okay, and that is what I have mentioned here. So evolution is a sequence of events which are involved into the development of a species or a taxonomic group of an organisms, right? So metabolic engineering, we do the same. Only thing is that we are going for the target, okay, to the specificity. So so the last line I can read, that metabolic engineering can be viewed as a method for in vitro evolution. Okay, कारण की आपण हे सगळे modifications कुठे करतोय? We are doing into a test tube. So we are doing into an laboratory grown cell. So we can call it as an in vitro evolution. Fine. So for going for an engineering into the cell, we require two components. So there are two components which we should take into consideration. One is called as an analytical component and the other is called as the synthetic component. Right. So now we are dividing the metabolic engineering in these two components, that is analytical and synthetic. Now what is this analytical and synthetic? Okay. So it means the other way you can call them as analysis and synthesis. So the synthetic component which is involved into the metabolic engineering, it first appears to be the molecular biology tools what you are using. Okay, means the different type of the methods, techniques used in molecular biology, genetic engineering, all those things, it composes the synthetic component of your metabolic engineering. Okay, so that is what I told you, that recombinant DNA technology. So that what we are doing, we are trying to go for manipulation of the gene. Okay, so either you may combine the genes, you may what you can say, delete a particular gene, anything can be done, right? And so where if you have the genetic background known for that organism, so it could be an E. coli, it could be the Arabidopsis, it could be an any of the human cell line or the insect cell line, etc., etc. So if you know the genetic background of that cell, you can easily go for the manipulation by using this recombinant DNA technology, right? So that is the synthetic component or the synthesis component of your metabolic engineering, right? So you can see here that you have the different models. So I have tried to show the different models. So it could be the salmonella, the mammalian cell line, the rice, E. coli. It could be the mus muscular, that is the rat. You can call it as the goat, Arabidopsis, Nicotiana, etc. Any system you can take. The cell of any system, maybe is having always is having the genes, and as we know that genes are the one which are actually deciding the overall functioning of the cell. So if you go into the modification of your genes, you may achieve the modification into your metabolism. And if you do the modification to the metabolism, you may get the different uh, products as an output, which you can see here. Fine. So these are arbitrary components which I have shown here just to give you and that we can have the output in which you are interested. Fine. So obviously you'll have to follow these four steps which you have already discussed, that is design, build, test, and learn. So doing all these kind of things, you can achieve the output in which you are interested. Now, what is the analytical component? So here in this case, the analytical component is nothing but the different type of the instrumentation what you are. So that's an engineering component of your metabolic engineering. Fine. So then, because there might be some questions which were raised, that how does one identify the targets for genetic engineering? Okay, that how do you come to know that which gene you have to target? So first you'll have to find out the metabolite, the reaction. Okay, then from that reaction you get the information about the enzyme, and from enzyme, as I told you, get the information of RNA, and then you come to the actually the gene. So for understanding that, what you'll have to study first, you'll have to study the metabolites, and that is nothing but the metabolomics what we are going to study. Now once you have studied the metabolite, you will try to study the reactions, and as we know, the reactions are catalyzed by whom? The enzymes. So you'll try to understand the what you can say, the different enzymology which is involved there. Okay, that which enzyme, what is the rate of the reaction, whether it is a regulatory enzyme, etc., etc. So all that kind of things you will study. Now we know enzymes are proteins, so obviously you'll have to study the proteomics part. And for studying the proteomics part, you may go for the Western blotting, you may go for the SDS page, you may go for 2D electrophoresis, you may go for the mass spectroscopy. Fine. So you get the information about the proteins. Now once you got the information of protein, from that you can get the information that what could be the RNA. So obviously you'll have to study the RNA component there, so that could constitute the transcriptomics. Okay, so the study of the RNAs is called as transcriptomics. So that is what the information you get, and once you get the information of RNA, you can find out what is the gene, and that is nothing but the genomics. Fine. So this is how you go for and for doing all these things, means metabolomics, the enzymology, the proteomics, the transcriptomics, the genomics, and for all these things you require a huge amount of instrumentation. So I already told you it could be the spectroscopic, chromatographic, or the microscopic instrument could be there. Like in case of genomics and no, no, yeah, genomics and transcriptomics, you may require the arrays, the microarrays you might have heard about. Fine. So you may require the microarrays. In case of the proteomics, you may require the mass spectroscopy or the 2D electrophoresis, which is there. So like this, the different uh, instruments. So few of the instruments which I have already shown here. So I think you got an idea that what is the engineering component. So it is nothing but the techniques or the instrument which has been used. Okay, and from that you will have to find out that which one is the accurate type of an technique where you get the wholesome information from that instrumentation, which can be then useful for you to decide upon whether to go for the no to decide upon that what should be the actual target for your metabolic manipulations. But as we know, the cellular metabolism is a complex network. Okay, so you have a very large complexity is there. So the complexity could be with respect to its genes. So you have a very huge genome for any given organism, and within that genome you have thousands and thousands of genes. Okay, but means as compared to the prokaryotic, eukaryotic, we know that they have a very huge genome. Not necessary that each and every gene within that genome is expressible. So so that kind of complexity is there. Now, within this genome also, you may have the variation into the expression of this genome. So the complexity, what I mean to say is with respect to all these things. Okay, that thousands of genes which are very, very tightly regulated. But these genes, they are responsible for carrying out this whole lot of complexity of the cellular metabolism. Fine. So you have many pathways which are occurring. Now these pathways, they are not independent, but all these pathways, they are dependent on each other. They work in well coordinated manner. So this indicates that there is a very tight regulation whereby this complex network is working within the cell. Okay, so this thing you should not underestimate. That is what I want to emphasize here, that the cellular metabolism is a very complex network, and within this complex network, if you try to do any kind of modification, just imagine what can happen, right? So that we should keep in mind. As I told you, that cellular metabolism is a complex interdependent network, and metabolome is a very small molecule of a cell. There are like this, there are thousands and thousands or lakhs of metabolites what you may have. Okay, and for that particular metabolites, you have a huge network of metabolic networks which are defined by some specific pathways. Okay, you may have to understand the flux. Now what is this flux? So flux is nothing but the rate of turnover of the molecules through a pathway. Okay, so again I can give an example of glucose 6 phosphate. Now glucose 6 phosphate is formed from glucose. So that reaction may be having a typical rate, that the moment the glucose is inside, glucose is converted into glucose 6 phosphate. So maybe at this point I can say that the rate is very high. But now this glucose 6 phosphate, it has a different fates. Okay, so either it can enter in glycolysis pathway, it can enter in glycogen synthesis, it can enter into the pentose phosphate pathway, etc. So the flux of this molecule or this metabolite is very important. So you'll have to understand the flux of that particular metabolite, that which pathway it is going through. Okay, and the last point which should be known, that is the regulation, that whether this flux of the metabolite through the different pathways, is it same or is it different? Okay, and is it that it is continuously undergoing all those particular pathways, or there is some or the other kind of regulation which is there, right? So these things also we should take into consideration when you are thinking of going for metabolic engineering. Now this metabolic engineering is like managing the traffic. So I think both these images you can compare with each other. Okay, so although I have not shown the whole complexity of the metabolic pathways, but this itself is showing you the complexity, and it can be compared with this the traffic what we have on the roads. Only the difference could be here, the carbons and the other atoms are the people here, metabolites are the location, and enzymes are the roads and railways that they travel upon. Because the nutrient which enters into the cell, it is in the form of carbon, hydrogen, nitrogen, oxygen. Okay, so these things which are entering, they can be called as the people. Now these carbon, nitrogen, oxygen, they are combined, and they are forming some typical precursors, some specific metabolites. Okay, so that is the route, that is the destination of those particular inorganic nutrients which are entering into it. And who is guiding them? Obviously it has to be the enzymes which are carrying out the actual reactions, right? So just try to imagine with these things, and the way you manage the traffic, the same way you will have to think about the uh, what you can say, the metabolic reactions which are occurring into the cell. Now the way we manage the traffic, that there has to be no overcrowding. The traffic should go smoothly. It should not be a continuous one-way type of an traffic. Always whenever there is an four-way road, जसं चौक असेल, so you should be able to manage the traffic, that only one way it should be on, something like that. So आपण ते कसं manage करतो, the same way you will have to think upon that if you are trying to do any kind of modification into the metabolic pathways, what kind of effect it will have onto the other pathways. So for that, I think this managing the traffic could be a very good example for it to understand the things. Yeah. So just look at this. I have tried to show a typical cell. Within that typical cell, there are some ingredients which are entering into the cell. Then that ingredients which has entered into the cell, or the nutrients which has entered into the cell, it is undergoing thousands of metabolic reactions into the metabolism, and at the end you can see some products around it. Now within this system, if you are thinking of a particular pathway that you want only one of the output to in higher concentration, so you will have to target that particular output, try to identify the pathway, and then try to see that are there any pathways which are in coordination with this typical pathways, and based on that you can go for the modification. Say suppose here you can see that you are interested in some product, say suppose here I say that we are interested in succinic acid, that succinate is required for some or the other purpose, so you are interested in succinic acid. So obviously succinic acid can be obtained from what? The sugars. So the sugars, it enter into the cell, it will undergo the glycolysis pathway, it will be converted into pyruvate. Next, pyruvate will be converted into acetyl CoA. Acetyl CoA will go into the TCA cycle, TCS, and from the TCA cycle you get the succinic acid. Okay, suppose I have identified this path, that I want this pathway to occur at a higher rate, and I want the succinic acid in higher concentration. But if you look here, there are many bifurcations. Like here, some intermediates of the glycolysis, they are going to glycerol. The pyruvate can be converted to acetaldehyde. It can go to the alpha acetoacetate. Then acetyl CoA, it can go for fatty acid synthesis. It can go for the synthesis of monoterpenes, acetic acid, etc., etc., etc. So there are many bifurcations. So this kind of complexity will have to take into consideration. Okay, and if you think of doing any modification, say suppose you say that I want succinic acid, I don't want this acetyl CoA to be converted to fatty acid CoA. So you may block this path, but then if you are blocking that path, the cell will not get the fatty acids which are required for its membrane. Okay, and if the fatty acids are not available, the cell may die. So what's the use? Okay, so you cannot be selfish at this particular point. So whenever you are going for any kind of engineering, you will have to think it as an wholesome, okay, as a wholesome system. Fine. So this is what we are going to study as an example. Then afterwards, making products of interest using biological systems as the machinery is the main aim of our metabolic engineering. Why? Because the wild types, they are not efficient, okay, or they are not able to produce your product of your interest in higher concentration. So that is one of the disadvantage of the wild type. Maybe they might be going at the drastic condition, but then you are not getting the product of your interest. And for this we use the bioengineering processes. Now by using these bioengineering processes, either you can upregulate the genomes, okay, or you can activate the genes, okay, so that the production can be obtained in a higher amount, or in some cases it can be you can suppress the genes so that a desired product cannot be produced. So any kind of modification can be done, and for doing this you'll have to go for modeling. Okay, so you'll have to prepare your own model where you are considered the all the type of the complexities which are involved into the cell. Okay, and you'll have to first lay down your hypothesis, that you can call it as a theoretical foundation. Okay, so that is what we do in build, that you first study the overall metabolism, okay, the complexity, then the proteins which are required, then the genes which are required, and the regulation of all this, everything we are trying to study into it, we lay down a theoretical foundation where it can be then studied by using the bioinformatics tools, and then we actually go for the experimental. Fine. So that is what maybe I may again explain you about the metabolism afterwards after this presentation. So that is what we'll have to do and then go for the experimental one. Fine. So keep this example in your mind, and we are trying to talk upon it. Fine. So if you are interested, the reference, the link I have mentioned here, you can refer this paper if you are interested into it. Otherwise, for your information, you can keep it like this. The next thing comes, what are the challenges if you're really thinking of going for the metabolic engineering? Because see, looking at the image, जसं आपण म्हणतो की गरगरायला होईल तुम्हाला. So look at the complexity. So it's really a challenge if you really think of going for metabolic engineering. Okay, that is one of the challenge. The second challenge could be the genetic elements which are involved for the expression of one particular gene. Like this, there are thousands of genes what we have in our body, in our genome. Okay, and each gene has its own genetic elements. So you may have to study all the genetic elements which are required. What are the genetic elements? Means the same thing which I told you. It could be the promoter, operator, etc. etc. Okay, now we are not going in the whole details of it. फक्त तुम्ही आता इथं कॉम्प्लेक्सिटी लक्षात घ्या की कुठे कुठे आहे. So पहिली कॉम्प्लेक्सिटी ही बघितली आपण, that they are interrelated to each other or they are connected to each other. Now the complexity could be also at the genetic level, right? And the third type of complexity, what you can say, the whole system, if you consider, there are many transport systems which are involved. Okay, there are many signaling mechanisms which are involved, because we know that these pathways, they do not work in one particular organelle. They are continuously moving from one compartment to the another compartment. Say for example, glucose is coming from outside to inside into the cytoplasm. So within the cytoplasm, it will be acted upon by the glycolysis pathway. Okay, now the end product of this glycolysis, that is the pyruvate, it cannot be converted into acetyl CoA or succinic acid in the cytoplasm. So for that, it has to go into the mitochondria. Okay, so once it has gone into the mitochondria, then it will be acted upon by the enzymes of your TCA cycle, and then you may get the succinic acid. Right. So the complexity is with respect to the different type of the what you can say, the transport mechanism, the signaling mechanism, then the different factors which are affecting the overall processes. So all those things you have to take into consideration. ठीक आहे, त्यामुळे हे एकदा आपण परत रिव्हिजिट करणार आहोत. आपण ऑलरेडी बऱ्यापैकी शिकलेलो आहे, but then I will taking you across the cellular metabolism in a different way to understand that what kind of complexity you may come across, or what kind of hurdles or challenges you may come across if you are thinking of going for the metabolic engineering. I think these pictures are self-explanatory. So what we try to ask ourselves is that, is there really a need of metabolic engineering? Yes, the question could be yes, because of the demand, the global demand. The living standards have varied. Okay, and initially only I told you that the chemical industry, what we are talking about, that chemical industry is not sufficient to provide us the required compounds in which you are interested. Okay, maybe if they are the simple molecules, you can easily synthesize them, but if you have a very complicated type of an molecule, it's very difficult to synthesize the chemical way. Fine. So that is one of the thing. And other problem which we can raise here, that if you are going for chemical synthesis, it is creating the environmental pollution. Okay, so this could be some of the disadvantages of the chemical industry, and that is why we are trying to work upon the biological system only. We are trying to modify or engineer the biological system so that whatever the product in which you are interested, you can get it in a higher amount. Okay, and that is to meet the need of the ever increasing population. Okay, so it could be the alcohol, what you get from the biological system. It could be the different type of the secondary metabolites, which we now know that they are having some pharmacological activities. So you can think of any kind of compound which can be easily obtained from this biological system. So that is why we can say that is one of the need of metabolic engineering. Okay, and that is what I have said here, that to overcome these challenges, the metabolic engineering provides the biotech industry with tools for rational strain design and optimization. So now we have modified the host, means we have the E. coli, which is an engineered E. coli, you can say. म्हणजे तो ई कोलाय असा आहे, इट्स तो एक रोबोटिक सिस्टीम प्रमाणे आपण त्याला तयार केलेला आहे की यू जस्ट कमांड हिम and that E. coli is going to produce whatever you want. Okay, so you can go for that kind of strain improvement, optimization of the nutrients, or the optimization of its growth, so that you get the desired product. Okay, so this brings about the significant shifts in manufacturing cost and the yields of desired products, which is suitable for the environment. Okay, so that can be one of the reason also in case of the chemical industry, that the cost may go on increasing. But if you are working with the E. coli, E. coli आपल्याला माहिती, it can be easily grown. It has a lifespan of around 24 hours. So within 24 hours, and by using some minimal media, which is very, very cheap, you can obtain your product in a very high concentration. So obviously the cost is going to be very low. Okay, so maybe the initial cost to develop the strain, etc., etc. तो एक component पकड़ला, you may have high investment, but once you have the system with you, the production can be, you can go for the continuous fermentation and you can have a huge production, and you can have the cost reduction in that case. Okay, परत एक एक्झाम्पल इथं तुम्हाला मी देऊ शकतो की इन द प्रेझेंट पॅन्डेमिक मे बी टू मॉडिफाय दी, तुम्हाला माहिती असेल आपण जे कोविशिल्ड वॅक्सिन आता घेतोय, दॅट कोविशिल्ड वॅक्सिन इज नथिंग बट अन चिंपांझी व्हायरस. जो आपण इंजेक्ट करतोय तो एक व्हायरस आहे, अँड विच इज अ चिंपांझी व्हायरस, अँड इन दॅट चिंपांझी व्हायरस वी हॅव ॲडेड दी जीन of the spikes protein of the coronavirus into this chimpanzee virus. Okay, and that we are now injecting. So maybe to have that modified chimpanzee virus with the gene of your spike proteins, the investment could have been very large. Okay, but now the production of that recombinant chimpanzee virus is very, very fast. Okay, and the cost is very low, and that is why I think so serum is ready to sell it at a very, very low cost. Okay, so maybe around ते काहीतरी 200 रुपयात वगैरे ते विकतायत. So why so less cost is there? Because of this what you can say, the biological system which we are using for the production. Fine. Yes, applications, it's very important to know that why do we go for the metabolic engineering, where it can have the different type of the applications. So it could be for environmental application, it may have, that is to generate the fuels from renewable resources. So I think you know what are renewable and non-renewable. So there is a need that you should have a fuels which can be renewed. Okay, then convert the biomass into chemicals. We have a huge biomass which is available. So it could be agriculture biomass, or it could be any other kind of the biomass which is simply going waste. So things has to work upon that, whereby you can convert this biomass into the useful products, or some cases you can use the therapeutic compounds, and that is what we are actually interested in. So for the production of the different secondary metabolites which are having some pharmacological applications. Fine, so this could be some of the applications, and now through this diagram you may come to know that n number of applications are there for metabolic engineering. That is in food ingredients to obtain the solvents, in cosmetics, fragrances, flavors, micropharma, pharmaceuticals, lubricants, polymers, fuels, etc., etc. Right? So these are some of the applications of the metabolic engineering which can be thought of. Okay, and there has been many agencies which are working onto it. Many industries have come up, and these industries or companies, they are being funded by many major institutes, funding agencies. So although the information which I could get is from other than Indian scenario. So it could be the US Department of Energy who has really worked upon it. Then it's the Bill Gates Foundation which is funding. Then there are some green alternative technologies which are coming up or which are mandatory for the petro industries. Okay, then so there are few companies which we can quote here, like Amyris is there, LS9 is there, Lygos is there, Genomatica is there. So these are the companies which have successfully worked upon metabolic engineering, or you can say that they have commercialized the products which are obtained through this metabolic engineering. Fine. So means it can be true for the Indian scenario also, where many funding agencies are there. It could be your AICT, CSIR, or the Department of Biotechnology is there, or Ayush Ministry is there. So these are some of the funding agencies which are also now promoting the Indian scientists or the Indian institutes to undergo this what you can say, the research on metabolic engineering, so that you can obtain many useful products from them. Right. So yes, so I think this picture will give you the idea. So see here, I have shown a native E. coli here. Now when you're using the native E. coli, the products what you get in which you are interested, so you may have the very low yield of that product. Okay, so you can think of modifying this E. coli by using the different uh, techniques of the metabolic engineering. So what kind of modifications can be done? So you can do a modification with respect to the glucose transport. You can modify the central metabolic pathway. You can modify the aromatic acid pathway. You can add a new branch also here. Okay, means you may have a system which is not producing an X product, but I will add some genes into it. I'll add new genes into it, so that new proteins will be produced and a new product can be obtained. So a branch can be added to it, or I can do the modification with respect to, like that E. coli is producing some toxic compound, and I don't want that toxic compound to be produced. So I may uh, introduce some pathway which will degrade that toxic compound within the system. Means the E. coli can produce the toxic compound, but I will add some what you can say, genes into it, so that the genes will produce the proteins which can degrade the toxic compound. I can modify the transport pathways also. You can modify the many signaling pathways also. So there are different ways and means where the modification can be done into the native E. coli. And once it has been modified, it can be converted into an engineered E. coli, what you see. And once you have the engineered E. coli, you can obtain the high yield of the product in which you are interested. Fine. And this is what is the metabolic engineering. Okay. So this 1, 2, 3, 4, 5, 6 which are given, they are giving you the idea that where you can think of going for modification. Yeah. So this is one of the company which I believe is very successful metabolic engineering, and as I told you that they have commercialized the products which are obtained through genetic engineering. Fine. So you can see the website, you can visit their website. It's really a good website. So if you are interested, you can visit it. So what they have done, they have converted the feedstock, the chemical feedstock, and they have been replaced by the petroleum based products. Right. So they have converted the sugar to BDO. BDO is one of their commercialized products. It's a trademark. BDO is what is a trademark, and using the microorganisms, they are trying to convert the sugar into the BDO. That is a petroleum product. It is one of the petroleum products. So they are producing around 30,000 tons of this renewable BDO per year, and this has increased the employment. It has helped to build the agriculture economy, etc., etc. Right. So from crop to cream means what they have done is they have created feedstock of the crops, that once you got the grains, the whatever is remaining, they have modified it, they have worked upon it, and they have tried to obtain the cream which can be used for your in your cosmetics, right? So then way the same way they have made some bio nylon, okay, and some polyamide intermediates are there. So this bio nylon, it has been used for what you can say, making the shoes or the clothes, okay. So in the textile industry it has been used, or you can see some of the polyamide intermediates are there which can have n number of applications for the humankind, fine. So some more examples which I have quoted, which about the products, renewable products which are being used, which has been produced by the metabolic engineering, right? So it could be lactate, acetate, succinate, so anaerobic, aerobic, and anaerobic fermentation, right? So if you look at here, the productivity was very high into the engineered organisms. So it could be ethanol, it could be the lactate, xylitol, alanine, etc., etc. Right? So although this could be the fermentative process, so these are the some of the examples which we can quote here. So if you look at this diagram which is shown here, this I got it from Current Opinion in Biotechnology. There was one research article in which this information is given. So you have that lignocellulose. Lignocellulose म्हणजे काय, जो चारा आपला राहतो शेतामधला. So that lignocellulose is being acted upon by some microorganisms, some modified microorganism, right? Fine. So this modified microorganism, they convert this lignocellulose into the ethanol, or it could be converting it into the liquid fuels, right? So that kind of work has been done, or one good example what you can take, that is the artemisinin, which I have told you, artemisinin, which is a very good antimicrobial compound, okay, but it is obtained in very very less amount, right? So they have tried to what you can say, produce this artemisinin into the E. coli, okay, and nowadays you have the engineered E. coli which can produce this artemisinin, so that nowadays they are available in the form of the tablets, and they have the commercially applications also, right? So these are some of the examples, or one more example which I can quote, that some around 8 to 10 years back we had scientist who has come from some American laboratory. He had visited our institute, and we had a guest lecture. So he gave an elaborative information about the sweet corn. So in the American or the European country, if you are aware of, what you can say, the corn is one of the most used, a staple food, I can say that it is one of the most commonly used food. Okay, so that particular corn, आता आपल्याकडे कॉर्न म्हटलं, मका म्हटलं की आपण डायरेक्ट खातो, ऍप्लिकेशन माहीत झाली की त्याचं पीठ करून आपण ते वापरायला लागलो. But those scientists, they have used this corn along with the modified microorganisms, maybe wild or the modified microorganism, and he gave us, or he showed us that products मिळवू शकतात. ओके, फक्त तुम्ही कंडिशन्स चेंज करा, थोडंफार मॉडिफिकेशन करा न्यूट्रिएंट्स मध्ये किंवा कशामध्ये, अँड यू कॅन गेट दी व्हरायटी ऑफ प्रॉडक्ट्स फ्रॉम द सिंगल raw material, right? And that is the what we can say, right? So I think with this, I think I have given you an idea about metabolomics and metabolic engineering. This was just an introductory thing which I have tried to explain. Now we'll be actually entering into the topic, what is metabolomics. Then we'll try to see that how to go for the modification. So the hypothesis which I told you, that how to lay down that particular hypothesis, what are the different ways and means whereby the modification can be done, and maybe at the end we may talk about some successful stories. Okay, so that is what uh, I'm going to tell you. Although it may require around some 4 to 5 lectures, I'm doubtful whether I'll be able to complete it on in that. पुढचं लेक्चर आहे का तुमचं?
हो सर.
लिंक आली का?
हो सर.
ओके.
अरे हा इतके दिवस घेत नव्हता, आता कसं काय घ्यायला लागला परत? ओके, तुम्हाला वेळ आहे नंतर परत कधी? Any suitable time which you can suggest where you will be available?